All right. So we've been asked to present the COVID Moonshot origin story. I'll take the start, and Alpha, can you move slides? Of course, from my perspective, the origin of this story is me sitting in my office reading newspapers. It was the beginning of February. And it was already clear back then that this pandemic that started in China is going to become global and is going to become a very important thing. That's why when Frank contacted me, the protease of the new coronavirus, and do we want to, should he send it over for us for screening? I didn't hesitate, and of course told him to send it as soon as possible. Me and Frank collaborated in the past on several screening projects. Our team at Weizmann is specialized at covalent screening, which is sort of a niche technique, but is very, very suitable for this exact target. Alpha, if you could progress the slide. So the target the COVID Moonshot project aims to inhibit is this Pac-Man you see over here, also known as the main protease, or MPRO, which has the role of, upon viral entry, the genome of the virus is translated into one long protein that is not functional in and of itself, and it needs this protease to process it to small functional units, these short colorful lines. And then the idea is, click, if we design an inhibitor that will block this protease, it will be unable to do its job, and then the virus will be unable to replicate in our cells. And to do that, we apply from the get-go was to develop a cheap and safe antiviral against SARS-CoV-2. Using the main protease as a target. And we wanted to do this faster than the traditional drug discovery pipeline because of the urgency of the pandemic. And then that's what led us to this open science crowdsourcing collaboration that sprawled until today.
So I'm Frank. For me, this project started when Martin was also on the call. Martin Walsh called me in when I walked past his office, and he said, Frank, we got this protease purified and we've got crystals of it. We've managed to crystallize it. So this animation, of course, shows the virus on the left and the protease itself. And I was quite stunned because I don't know how his team did it this fast. And I said, well, of course, we have to quickly do see if we can get a fragment screen, because he said they already had crystals that were suitable for fragment screening. We should add, this can be quite a big challenge. So the facility we're at Diamond is all about shooting compounds into crystals. Now, sorry, this animation is going a bit faster than I thought, is basically trying to show you the crystals. You need to be able to get protein crystals that are, of which you can grow hundreds of them in these little drops, which flashed by a minute ago. And then the facility, which we've called XChem, will add compounds, fragment compounds, small compounds to each crystal. And then they sit there and soak for maybe a few hours. And then you have to pick up all these crystals one by one and put them, freeze them into liquid nitrogen. And then the whole beamline, the whole setup will add them one by one into this diffractometer, which you see in front of you. There's the diffraction pattern that gets measured. And we do hundreds of these diffraction patterns. In this case, we did more than 1,500 of them. And by the end, each diffraction pattern is assembled through complex mathematics and data processing into the three-dimensional structure of the protein. And for a small set of those, you will see in that the protein structure evidence of the compound that's bound and how exactly it has bound. Because it is a three-dimensional view of it, it's a great starting point for designing compounds. You don't have to guess what sort of compounds you want to make. You can actually design them almost from the screen. And so this is the timeline. I mean, I think Martin's group got the DNA into the lab by middle of February, and by a few days later, actually, they already had the protein structures. And while Daren was doing their electrophile screening and Weizmann, we were doing the crystal-based fragment screen. And the output of that was this superposition. We had about 70 fragments in total that we observed in the binding site. And we were sitting and thinking what to do with it. In fact, we'd submitted a grant proposal to the Medical Research Council here. It was out of scope. People didn't want at the time to fund novel targets. You can progress one, please. But I should say what this speed of progressing was for us quite staggering. It was a massive team effort between Martin and my teams and also Near's to generate all this data. And then we just put it out. Could you just put the next slide? Put it out in the public domain. And
yeah, so that's when
Alpha.
Right. So that's when the team at PostEra entered this project. We saw the beautiful data that Frank and Martin and Nia posted on Twitter. And when we looked at the structure, back then we were in San Francisco, starting PostEra up, we realized that there's a lot of very beautiful overlaps, fragment overlaps in the main protease binding site. And we thought, would it be great if we can turn these fragments into a drug using machine learning technologies, which we have been developing over the past few years. And so it all started with a tweet. We saw Martin's really nice tweet about releasing all the data. And then we tweeted back mentioning our technology in synthesis planning and fragment elaboration. And lo and behold, after a few iterations, we quickly jumped on a Zoom call and realized perhaps the best way to kickstart this project is why not just releasing all the fragments out there, and everyone's working from home in March last year. Why not just launch a crowdsourcing challenge where we literally ask people to literally submit compounds to our crowdsourcing portal, look at the fragments, submit compounds which people deem to recapitulate fragment mergers, and then we would triage these compounds using ML, machine learning, and synthesis prioritization, and we'll make the compounds that are easy to make, just as a first pass to go from fragment to hit. And Nia tweeted this website and crowdsourcing portal out, and within we thought, fragment merging is fun, but it's not a video game, so we might get maybe a few dozen people signing up, but we got a lot of traction, really humbled by the excitement that we have received from the community. People are submitting a lot of compound design using a lot of different methods. And so we triaged these compounds just in the first round synthetic accessibility, actually, and make as much as we can just to test out all these wonderful hypotheses. And it started off with a few people on a Zoom call. It blossomed into a global collaboration spanning literally every part of the world, with people offering in-kind contributions in terms of biochemical assays, in terms of experimental resources. And we are to this day hugely indebted to all our wonderful collaborators, which we have met in this journey. To date, 20,000 unique compounds. We made and tested 2,200 compounds and found 400 potent ones, sub-nanomolar. And a quick tour of the progress. Ed will tell you a lot more about Medchem. We started off as a fragment-based design project with fragments obtained by Frank and Martin and Nia, quickly merged these fragments into a 24 micromolar hit. This is actually a very synthetic facile molecule, one-step synthesis, going to 720 nanomolar, again, still keeping it very synthetically facile, and then evolving this to a benzopyran, jumped up the potency down to 140 nanomolar. This is still a one-step synthesis, and we were able to keep progress pretty quick, design make test cycle quick, by focusing on rapid synthesis as a way to actuate design. So even the third compound here is still a one-step coupling, and all the way down to 80 nanomolar compound as our lead, with facile synthetic route and a strong antiviral activity. And throughout the campaign, we use machine learning as a way to keep track of synthetic complexity. And we actually show that algorithms can predict the wall clock, the predicted synthetic complexity of compounds, track the wall clock time of synthesis, allowing the design team to prioritize compounds in a way that is efficient. And in the journey, we've also developed machine learning technologies for fragment merging and rapid hit-to-lead by structural biology informed parallel library design. So using ML to turn high throughput structural biology into predictive inhibitor design.
So I'm John Chodera. I'm at the Memorial Sloan Kettering Cancer Center, and I we were incredibly lucky early on in the project to recruit Ed Griffen to lead the medicinal chemistry team. And he'll tell you more about his excellent work leading that team a little bit later. But I had been working with Frank van Delft earlier before COVID. And one of the great resources our lab has is Folding at Home, a worldwide distributed computing resource where hundreds of thousands of people around the world contribute their computing power, often gaming computers with GPUs, in order to do molecular simulations that we thought we could harness to help us progress these fragments towards potency. And so we were able to get the Folding at Home Consortium, which consists of multiple laboratories, and especially with help from Vincent Vols's lab at Temple University and our own laboratory at MSK, we managed to leverage the amazing initiative from all of these folks that had started to volunteer their computing time. We quickly had 1 million people volunteer over the course of a few months in the pandemic, bringing us past the exaflops barrier and making us the most powerful distributed supercomputer in the world. And we were able to leverage this for several different stages of this fantastic effort, both helping prioritize some of the early stage molecules by absolute potency through absolute free energy calculations, and then later through these sprints, which are still continuing, to help us identify which of the many, many thousands of compounds we could synthesize thanks to Enamine's fantastic building block technologies would be the most potent to synthesize and pursue. This is just a representation of where everyone all over the world has contributed. Alpha, if you can advance the next slide. This allowed us, together with these great teams, all the volunteers, to start from where we had seen before with this fragment screen in March 2020 to make extremely rapid progress, as Alpha has hinted at. We had very rapid fragment mergers that got us on the map. We got first nanomolar inhibitors very quickly in July, and then we got good potency in antiviral activity in antiviral assays. And then very rapidly, this took us all the way to great compounds that were very potent leads that had great antiviral activity and good selectivity against host proteases. And we were excited to finally be funded to take this all the way through preclinical studies to clinical trial readiness through a Wellcome Trust grant. We'll hear more about the strategy for that shortly. This only took us about $1 million and about 18 months to get to this process of late stage leads. It's remarkably fast and remarkably cheap for a drug discovery effort. And the compounds, the great variety of them, along with backup series that have been generated, offer us a lot of hope that we'll be able to have non-covalent compounds that might offer complementary approaches to the existing routes for therapeutics, especially for low and middle income countries. And like I said, we'll hear more about that shortly. Next slide. We did learn a lot during this process. First of all, you need to invest in pandemic preparedness. There's no shortcuts for these. It just requires spending the money that you need to be prepared. It's not a lot of money in the grand scheme of things compared to the cost of the pandemic in lives and in lost economy dollars. It's a very, very minuscule cost. Thankfully, some governments are finally starting to realize that this is important, and we can't leave this to the capitalistic enterprise necessarily, because there's not a profit incentive for saving hundreds of millions of lives if you have to prepare, and if there's no guarantee you'll make the money back in the lifetime of a patent, for example. What's great, though, is that it does seem like modern drug discovery technologies, much like we've seen vaccine technologies have been accelerated, they really are up to the task, and they can field very accelerated efforts that can happen in time to actually impact the pandemic. The other great thing is that we have shown that non-corporate initiatives, especially working with great nonprofit folks like the Drugs for Neglected Diseases Initiative, DNDi, are really able to complement commercial drug discovery and provide useful utility and hopefully for us, a patent-free drug that's available at very low cost for the global community. And it's important that we realize we can't think about this locally. As we've seen from the emergence of variants across the world, it's not like we can keep anything out. We're a global community, and the spread of variants and pandemics is global. And so we're not safe until everyone is safe. It's important to keep in mind. Next. Well, thank you. And we'll now pass it over to the next stage. I should use this opportunity to say that if you do have any questions, please put them in the chat and we'll address them at the end of the Q&A.
I'm Ed Griffen, so I'm going to be taking you through the medicinal chemistry story of the COVID Moonshot over the last year. Just let me get that sharing properly. Okay, two very important slides that I'm just going to start off with. First of all, I'm going to show you real active antivirals, and some of those have oral bioavailability. So on the front of our website, there's this very important statement. These are compounds that have not been tested fully for toxic activity. Everybody here, I'm presuming, is a grown-up. You don't go away and make these and expect them to work for you, but we have to put that up because this is getting fairly serious. So because we haven't done toxicity testing, these compounds are still at risk. So don't go away and make them and try them on yourself. Second really important slide is I'm here representing a brilliant team of people. These folk represent the design team. There are a huge number more people who've contributed individual designs. These are folk who come up regularly, review those, provide their expert input. I'm hugely grateful to all of them for everything they've done over the last year. There are more than 130 other people who've worked on the project. It grows almost daily. It's referenced in a bioarchive that just grows like Topsy. We've also had some amazing in-kind contributions. So UCB, CDD, who provide our data store, Novartis, who provide some outstanding screening capabilities for us, and Takeda, all of whom have provided a lot of in-kind contribution. I'm going to take you through a year's worth of medicinal chemistry, the thick end of 2,000 compounds, and I'm going to try and do it in 15 minutes. Honestly, I've never done that before, so it's going to be a bit of a journey for all of us. Our clinical ambition is really clear. We want to make the right kind of antiviral. So we want a compound that is extremely potent and safe, but it's got to act here in the early stage of the disease. Most of us know enough about COVID now, unfortunately, to know that you've got to get in early while it's in the viral stage before you get severe illness and critical illness, which is where you end up in hospital. And because we want to go early, we're going to be used in the clinic. That means it's got to be safe for everybody, and it's got to be a tablet you can take. In the limit, you might even consider being able to use it for post-exposure prophylaxis, but at least when you're confirmed that you've got COVID, you could take it. It would suppress your viral exposure and stop you ending up in hospital. And that covers all sorts of the population. So obviously the very at risk and those who have been either are non-vaccinated because it didn't work because of other underlying health conditions, or even the vaccine hesitant. Any of them could be treated. In the end, we're all about reducing hospitalization and mortality for anybody who might be exposed. This defines what kind of drug we want to have, though. And so we translate that aspiration into a checklist of things that we call a medicinal chemistry target product profile. So we know what the enzyme is. We've had the picture of the little Pac-Man. We need to inhibit that. But more importantly, we actually have to inhibit viral replication in cells. In terms of how that works, in terms of pharmacodynamics, pharmacokinetics, we want to have the blood levels of the compound suppressing the virus for 24 hours. That's our base case assumption. We want to basically hit all variants. We are focused on SARS-CoV-2. If it hit other coronaviruses, that's an additional benefit, but that's not our central goal. We must have an oral agent. We want decent half-life in human. Okay, maybe if you had to take it 3 times a day, that will be okay. But obviously twice a day, once a day will be perfect. And it's got to be very safe because we're looking at treating an entire population of anybody from whether they are male, female, whatever age, it doesn't matter. Okay, let's move on. Right, so design principles. I'm not sure where this line's come from. We're aiming for small, efficient molecules, less opportunity for less off-target effects. We want to reduce our permeability and metabolic risks, as I've just talked about, because we want to have maximum exposure. We also think about the substrate envelope to minimize resistance costs, and try and keep the compounds simple if we can. And that reduces the development cost of goods, which means faster, and therefore we start dealing with the pandemic quicker. In our case, we're trying to avoid peptide and mimetics because we just want to present a different development and toxicity risk profile. There is nothing wrong with these in principle. It's just a lot of other people are working in that space already, and we're trying to do something that will be complementary. We may end up with covalent compounds, but our strategy was get the compounds potent and selective first, add a covalent warhead if needed. So that's an efficient selective agent rather than just a hot warhead. I'm not going to talk about the pan-coronas strategy because that's not really a part of this program. Okay, let's try and get everything in one molecule. That's the key goal over the last year. So we want cellular potency, oral bioavailability, and safety. And the things that contribute to that are basically enzyme potency and permeability for cellular potency, solubility absorption and low first pass clearance for oral bioavailability, and for safety, don't hit anything that's an off target and avoid drug-drug interactions. As I said, we may be using working geriatric patients, so we don't want to be, if people are taking other medications, we don't want that to be an issue. Okay, and some of those are known risks like cardiac ion channels, genotoxicity, CYP450 is key in terms of drug-drug interactions, but we can also go looking for other critical targets, and we use the SERP44 panel for that. Okay, how we do this in practice is we have an assay cascade, so we screen against the enzyme itself. We use a fluorescence assay that's run at the Weizmann, and we use rapid fire assay that is run in Oxford. We look at solubility, we do structures at Diamond, and then we go into in vivo testing and we look at exposure in rodents, often as racemates. I'll show you some of the compounds and explain why. And in parallel, we do antiviral cell assays, and we have an outstanding, even broader set than this of collaborators who've run those antiviral cell assays, which are hard assays to run in BSL-3 facilities, and we're hugely grateful for them for providing this absolutely essential data. Selected examples, we look for permeability, and I'll talk a little bit about that later on. Rat and human protein binding and safety studies. We then go into, we started off looking at mouse, and we now predominantly look at rat, and so then we can predict our human doses, and we have started doing human pharmacokinetic prediction to make sure that when we nominate a compound, it stands a good chance of actually showing the right exposure in people. Okay, we've already had this view of what we achieved last year, and basically what we got to with it incredibly quickly was compounds like this, potent with a really good spectrum. So I'm just recapitulating that we had good cell potency, good enzyme potency, at least on the board for cell potency, no cytotoxicity, really good selectivity, good permeability, and we're starting to be at least measure human liver clearance, but this is nowhere near good enough. But we're getting some rat oral bioavailability. So at least as a starting point, these were phenomenally good start points. They're small, they're potent, and they do at least show some oral bioavailability. And what we've done over the last year is we've built that. And here I'm just showing a selection of steps where you can see everything I show in terms of picture, that's real, that's not a model, that's out of the Diamond structure facility. Here you can see really straightforward medchem. We did just a methyl group scan and picked out that this compound, it's weaker, but you can put a methyl group here. And the hypothesis was we would bias the conformation of this amide up into this P1 pocket. And that kind of worked, but it's not desperately good, really. However, one of the contributors, Pete Kenny, looked at this and said, well, actually, why don't we try just extending that slightly with a methoxy? Because that means we could reach across into the S1 prime subsite. Now, it turns out that second part's really difficult, but this just methoxy substitution here is enough to basically double the potency while reducing the lipophilicity. So this was a great addition, this compound. When we profile that, it's a pretty good looking compound. So it's antiviral, it's soluble. Yes, the human liver clearance is a little bit high. That's not really where we want it to be, but we've still got exposure in the rat. So as a starting point, not bad at all. And we looked at it in terms of in vitro exposure. This is in mouse, oral exposure, we're getting a little, and the problem is really not enough. And even if you try significantly increasing the dose, you're not seeing enough cover. Oh, so the compound is not staying in the blood long enough at a high enough level to really cover the cellular potency. So that combination of cover and potency isn't in the right place with this compound, but nevertheless, it was a great starting point. We carry on doing, amongst all the other things, really good standard medicinal chemistry. And one of the things that my colleague Ralph at Thames Pharma Partners looked at was just systematically exploring the SAR around this ring. And again, this very straightforward shift from chloro to difluoro gives a small improvement in potency. But if you could think of, okay, so that's kind of interesting, but let's take that with the previous learning of cyclizing around here and adding the methoxy. And that gives a compound that is, roughly speaking, the same. And to be honest, this compound languished for a bit because it wasn't spectacularly better. But what was interesting, as we further profiled it, it turned out that it was significantly better in terms of antiviral potency. It's actually significantly better in terms of solubility. It's made a real step forward, so it's roughly twice as stable in human liver hepatocytes, which is great. And also in terms of bioavailability, it's about, you get about twice as much exposure from an oral dose. So all told, despite the fact that the enzyme activity is actually worse, it's about twofold worse, in terms of how you do in terms of getting that exposure over the antiviral effect, we've made a great step forward. So you might be asking at this point, well, why do you care so much about rats? Well, we do the lead optimization phase in the lab, but what we've got to do is look forward all the way to getting to patients. And if you think about preclinical development, you've got to check the compounds are safe in animal models before you can get to people. And typically you do that in rat and dog. And from that, you can also, you're going to be predicting your human dose levels. So to get to the clinical development, to get to patients, you have to go through rat. And if you can't go through a preclinical species and demonstrate safety, you won't be able to go into clinical development. So that's why we care about getting these good rodent levels. So what happens if we actually take this compound through? And I'm very grateful to Gwen Fate, who's worked on this project with us, for doing these predictions. So here's now a whole mass of in vitro testing data. So this is human, rat, dog microsomes, which we can use for making a human prediction. Here's the measured in IV pharmacokinetics and the oral pharmacokinetics. And from all of this, you can start to actually generate a prediction of what would this compound be like if we were to dose it to people, given we know about the pharmacokinetics and we know about the cellular potency. And we can edge towards a first prediction of how much compound would it take to use this as actually an antiviral. And the problem is that because it's still relatively rapidly cleared, that predicted dose is somewhere between 300 and 3,000 milligrams 3 times a day. And that's just too high a dose. So this is not a practical anti-COVID agent. So we can't take this one forward yet because we can find better compounds. But we've learned a huge amount in even generating this. So our next question is, well, is this ring the problem? And we generated a whole series of compounds. All that varies is X1, X2, X3. So there's this table showing how we just permutate the different rings. This is very classic, thorough medicinal chemistry. And what we discovered was that you can actually tolerate polarity here, which is not immediately obvious from the crystal structure. So that's why you have to go away and do the experiment. And you can look if the tetrahydroisoquinoline, so in other words, where we have nitrogen in this position, as long as that's not basic, it looks quite promising. When we look at designs, and hundreds, thousands of people have contributed to design, there's really a division. Sometimes people think they can design too well, and molecules come out a bit complex, or they don't take account of the SAR. And that's kind of upsetting when that happens, because we already know so much. I'm going to have to speed up because I'm already over time. This was a great piece of design. We learned a lot. We turned around compounds very quickly, but we made a scanning library looking for interaction with glycine 189. And we actually were able to push our potency all the way down to 70 nanomolar in this case, where hopefully now we've stabilized the compound compared to the benzopyran. So this was a hugely successful piece of work. Of course, it also shows you that you should test your null hypothesis, because allegedly this is still basic, and the prior data said that that wouldn't work, which is kind of interesting. These compounds are great. They're more potent. They now have longer half-lives, particularly in this case. They don't hit glycine 189. How they generate their potency is still of great interest to us. We've done more in vivo profiling. This is a great looking compound. In terms of antiviral efficacy, we've now taken another step forward in terms of clearance. They're more slowly cleared. They look great in human. However, the rat half-life is still too low and the bioavailability is still too low, but we are making progress. In terms of antiviral effect, if you look across multiple cell lines, this is Paxlovid, which Pfizer are launching, I believe, in January. We have got potency that is equivalent to that in cell lines. We haven't got the same PK yet, but we're making good progress. So I said we need everything in one molecule. This is all about how we've understood the permeability using data that Novartis have helped us generate to predict how we get into cells. We can say that when the compounds are potent, so this is the Weizmann enzyme assay, and they are permeable, you get into the happy green zone in the top right of high cellular activity, and that's outstanding. We can actually use that to generate actual cutoffs for how we design our compounds. So where we are now, we've discovered that one of the things we can do is we can spirocyclize here, and that leads to another decrease in terms of clearance, which is great. So that was the compound I just showed you. You spirocyclize it, it gets more stable, the free drug goes up, looking like a very exciting compound. We can generate compounds with even better bioavailability. So this is 98% bioavailable, slightly different, but you can see where our thinking might be with this. We can generate compounds with good long half-lives. So we're up at 5-hour half-life in rats. So we know it's doable. We just don't have the potency in this. So it's trying to get all the properties right in one compound ready to nominate. I wanted to highlight some of the outstanding chemistry that's been done in the project. This is literally taken from this Tuesday's update from our colleagues at Enamine, and we don't just do simple chemistry anymore. So this is a great synthetic sequence. So in order to make these analogs with cyclic sultams, this is really clever chemistry. So making the sulfonamide is straightforward, then an intramolecular HEC cleave these rings, and that liberates this acid. And then if that wasn't cool enough, you can then form the anion here, quench it. You form the anion here, quench it, reduce that ring system, and it cyclizes. And then you can sew on the isoquinoline here, which is the P1 binding group, a couple of straight deprotection and binding, and you generate these spiro compounds really clever. This is clever synthetic chemistry applied to making great new inhibitors. Are we going to get there? That space where you have cellular potency, oral bioavailability, and safety is small, but it does look like that's where our preclinical compounds will lie. Thank you very much indeed. I hope this was helpful, and I'll pass on to the next speaker.
I think you just need to stop. Okay, thanks. Hi. So this is the moonshot view from the trenches. Admittedly, I've been in quite a few trenches on this project, so this may become more of a high-level overview, but I'm part of PostEra. What I'm going to talk about today is more of the moonshot infrastructure, but I've also been involved as a scientist on the project. All right, so just kind of another side of the story going back to March 2020. Obviously, there was this desire to run a crowdsourcing experiment based on the initial fragments. And there was a lot of discussion as to how to do this. You know, you could use Google Sheets, email, survey software. Having worked, you know, on some projects where things are exchanged by Excel, I think a few of us knew this was not going to work. Just as a description, just to get the author list for the moonshot paper, which just asks for a name, an email, and a title, I had over 30 emails asking about edits to the Google Sheet. So we knew we needed some way to represent the much more complicated chemical data. So, you know, we knew we needed an app where people could easily input structures and their designs. So an early version of this web app was built in one highly caffeinated weekend by me and another programmer on the team. So going back past my chemistry to some of my web development experience. And I've taken the screenshot of the site as it is now at postera.ai/COVID because it is generally the same interface. You enter a structure and enter your rationale. As I think Alpha mentioned, we were expecting a couple structures, quickly overloaded the site with tons of submissions. So this is also a graph of my resting heart rate as we were trying to fix all of the bugs that you all were reporting and kind of trying to get this project off the ground live. But, you know, actually, we did successfully get a lot of submissions. So this was an early submission from the crowdsourcing initiative. You can see it has here what it's inspired by, what the design rationale is, and even the availability. And this was a starting point for a lot of the further development. So once that all began, we actually then had to figure out how to manage this whole project. So, you know, in an ideal world, drug discovery projects often have synthesis and experiments at the same site or nearby. You're reducing the logistics coordination that has to be done. As this graph has shown earlier, that is not this project. This is an incredibly distributed team. I've actually only met, I think, 3 of the 130 authors on the project in person ever. So in an open science project during global lockdown, depending on assays that didn't really exist yet in some places, in some instances, you have to figure out a way to do this highly distributed. We did get some early help from the great folks at CDD who could really help out with our biological standardization. And so every design is now linked to the biological data created by it, created for it in CDD, and also linked to a manifold page where you can do a lot of the analysis of the speed, especially early on. So just stepping back, realizing what we needed to really get this going quickly. We needed an easy way to leverage the large catalogs from places like Enamine and order lots of purchasable molecules. We also needed to understand the inventory of every CRO we were working with, which I think we've now worked with 4 different synthesis CROs at the same time in some instances. You also need accurate prediction of the synthetic difficulty as you are getting these large amounts of design. And then further, you also need ways to filter out the bad designs. Early on, we did get some submissions that were for things such as soap molecules or Schedule 1 drugs. So, you know, we did have to find the best of the crowdsourced submissions out there. So actually, what I can show you just really quickly is that this now, which was developed for this project, is a tool called Manifold, which is actually free to all Moonshot collaborators and academics still. And this was actually an example query from the project. So a library built or designed by synthetic chemists, medicinal chemists, machine learning engineers, computational chemists. And it's been ranked by the programmatic API to tell us, hey, what can we get off the shelf within 3 weeks to get to the assay? So at the top is going to be those compounds where we can make and test them very quickly. And then at the bottom are the ones that we can start to understand will be a significant investment. And you can always click in to see, hey, why is this one actually ranked highly? Can we get the building blocks? Actually, this is how it was made on the project, I believe, this exact route. So it just allows us to answer questions like that, and especially at the hit to lead stage, allowed us to go super quickly, understand what was available at our friends at Enamine. We knew every time, hey, we can get this within 3 weeks and try this molecule quickly. So that's kind of main overview I wanted to say is, you know, a lot of infrastructure has been built for this project. Some of it is actually now a product that's used in multiple continents on other types of drug discovery projects by commercial entities. And a lot of it has allowed this project to go very quickly and remain so distributed. So, you know, we can all benefit from the tools that are developed even to help out these antiviral projects. So you can visit that at Manifold. You can always talk to me about any of the other ways we've structured things, how we've organized these projects. I'm always open for email, for a chat. So just want to say thank you to both PostEra and Moonshot teams. One day I will eventually meet most of you and get a picture, but it's been a pleasure being on this project. And I'll hand over to Slava, who can talk about the great work at Enamine.
Hi, everyone. Many thanks for the introduction, Matt. So I will talk about Enamine, what we do, and how we were so lucky to become an organic part of the COVID initiative, and why and when. We are a contract research organization, basically. We are doing chemistry, lots of chemistry. But we are a private company located in Kyiv, Ukraine. We are 30 years old, and we currently employ over 700 organic chemists, which is our main asset. We do not have our own drug research. We position ourselves as a service company, and at the same time, I would dare say we are an unusual CRO because on top of the contract services, we are also a vendor. We have a catalog of building blocks and screening compounds accessible for the scientific community. And also we are educational platform because we do not hire our chemists, we teach them. So from the bench and from the university, we teach our people, we teach our chemists in our labs with hopes that most of them will stay to work with us. As for the medicinal chemistry and custom synthesis, why it was so important when the COVID hit and the consortium assembled at the same time, the worldwide logistics were collapsed. And we were very lucky to have here in place all the reagents readily available from the shelf. And from those building blocks, having them, you can easily have in 1, 2, 3 steps reaction, or in one pot reaction, many screening compounds that can be made quite fast and very effectively, as the validated parallel chemistry protocols are also already known. And this concept, some of them may already known as a real database concept that we are trying to develop in order to produce and synthesize novel compounds very fast and with high effectivity. Still, our main assets are our people, our experienced chemists that we have here in-house, because not everything you can make by parallel chemistry, and sometimes when the lab work is required and bench chemist is involved. So we do have those fantastic chemists who have and working with a variety of instruments. And here is our, as we say, tools of the trade, because we do have a dedicated lab for example, carbonylation, fluorination, ozonolysis, flow chemistry, and this COVID initiative was the first advantage. We had everything here in place, the building blocks and the reagents. Then we had this amazing facility where we can separate enantiomers to perform reverse phase preparative HPLC and SFC. And at the same time, we still managed on the rocket speed to make those compounds and to deliver them just within a few days after they've been synthesized to the variety of destinations worldwide. So our logistics and compound management is managing this so far. It was also very challenging to align the resources, the human resources, to the project stage. Sometimes it was, for example, to progressing for hit to lead, where more or less chemists needs to be involved. So there were a time when we had from 4 to 20 people, FTE chemists involved in different stages of the project. And we were happy and flexibly accepting those challenges. And he's our amazing winning FTE team. So, and I would like to, at first instance, thanks to those people who actually made this chemistry happen. And thanks to the COVID Moonshot, and thanks to everyone. And it was another amazing year for us at Enamine to be honored to work with COVID initiative. And we are very much looking forward to the next year and next Medchem challenges. Many thanks. And let me also take this opportunity to introduce the next speaker, who is Haim Barr from Weizmann University of Science, who is heading the drug discovery.
Hi, yes, thank you very much. Thanks for the opportunity to give you a tour of the kitchen. Perhaps never a good idea to look at the kitchen in your favorite restaurant, but this is where we're doing the protease screening assays in the Israel National Center for Personalized Medicine. So first, I'll give you a shout out to the wonderful people that are doing the work and involved, the people from Near London's group and the people from my group on the left. So a lot of the reagents were developed by Ronen and Ram and Near's group, the substrates, and some of the initial assay development by Fearon and Annette. And as you heard from Diamond, Martin Welsh's group makes the main protease that we use in the assays. On our side, Galit is our compound manager, and Hadir is our assay technician, who's making the majority of the data, which is driving the screening assay. So back in May of 2020, Near Ronen and Fearon delivered us an early version of the main protease assay. We did, as screeners, we always want to do a little bit more optimization and miniaturization in order to adapt the assay for an automated screening platform, including a counter assay to eliminate potential interference compounds, which later on, as the project developed, really seemed to be quite superfluous because most of the compounds are really devoid of any kind of interference. Initially, we worked in a screening mode for about 13 rounds. The compounds that were coming in were tested in 2 concentrations. The active compounds then went through dose response and a counter assay. And then since about August 2020, we've been going straight ahead into 13-point dose response, doing 3-fold dilutions. This covers a large concentration range, and it's particularly informative. In the event that we get compounds that are more potent, then we move down a phase to 10-fold lower. So this has been driving the same process that Ed alluded to and Alpha alluded to, the design, make, assay, and interpret process. In currently 86 rounds and counting since the inception of the project. And we've already seen the wonderful progress, how it appears weekly in CDD. So it sort of, how does it happen from my perspective? At some point during the week, I'm going to get an email from Enamine that the shipment of compounds are on the way. Matt is going to register the compounds in CDD, and then they're going to arrive at our department, where we're going to register them in our internal inventory tracking system. We're going to move the compound, or Galit, I should say, is going to move the compounds into acoustic dispense plates and then split off whatever's left over for future use. Galit then will generate assay-ready dose response plates and also a mapping file of where the compound is located in the plates. And the assay will hand it off to Hadir, who's going to do the assay sometime in the next couple of days following the preparation of the plates. And then it's going to come back to me for data normalization and then publishing to the rest of the Moonshot community via CDD. If needed, and earlier on the project, many of the compounds were then going over to the IIBR for viral assay, but we already have accumulated quite a lot of compound which can be used if needed. The way the compounds, the way it looks on the deck, as we say, you see on the left-hand side, the standardized template for compound positions in our plate. We're going to put them in sort of a staggered format, so it minimizes trending that you might have from the automated type of assay execution. And on the right-hand side, you see the acoustic dispensing instrumentation. The assay, Nir described the assay last year, but just to remind you, it's a fluorescence assay. It's a quenched peptide that upon cleavage by the Pac-Man main protease, we get a free fluorescinated product, which is fluorescent. And then you can see down on the right-hand side of the screen, the way the heat map of inhibition maps over to the compounds. The compound would be typically put in 1.5 columns, as I said, in a kind of staggered format. And then the data, which I'm going to upload to CDD, would then be looked at and sort of annotated. Perhaps sometimes there might be some individual points that need to be removed, or is some sort of additional tweaking of the dose response curve. We also manage the project through a sort of electronic project management platform as well for sort of scheduling our tasks and communicating within the team, and as well using an ELN to accumulate all the data and all the documentation. So just to kind of finalize why it works so well, I think we're using a really well-validated target. We have the incredibly good reagents and a robust assay, and just all the phenomenal collaborators on the project in the sense of team spirit. So with that, I'll pass over to the next speaker, and I wish everybody success in 2022, more success. Daren.
Hey everyone, I'm Daren. I work at Diamond Light Source. For you that aren't aware of Diamond, we're the UK's national synchrotron, and we're based roughly 15 miles just south of Oxford in the countryside here. And I'm responsible for looking after the XChem fragment screening platform, which is a high throughput crystallographic fragment screening platform. I'm not going to go into details of the different components of our platform. I just want to kind of highlight the general workflow we have for going from crystals to actual dissemination of fragment structures. We're a well-established workflow, which routinely we can carry out screens of up to 1,000 samples in terms of data collection and sample preparation in about a week. But for the whole process, from moving from crystals to actual structures, we can typically do about 1,000 samples in 2 to 3 weeks. We've been operating now for about 5-6 years, so we're quite well established. And this meant that when Martin Walsh chapped on Frank's door with the crystals of the main protease, we were quite well positioned to carry out a fragment screen against this target.
So I appreciate that the title of the section of the talk is the Moonshot trenches, but we've been in a few trenches for the last year or 2 years in the XChem team. So been working pretty continuously on various different code projects, as well as our targets across the last 18 to 24 months, starting off with main protease in February 2020. And we did have a short period where we had to suspend our user program for various different reasons, but otherwise we've been running continuously, screening our fragment libraries against 7 different COVID targets, as well as maintaining our user program for the majority of the last 18 months. But alongside all of this, we've been providing continuous structural support to the COVID Moonshot. So we've collected over 6,000 data sets to date. So I just want to summarize some of the stats that we've actually achieved over the last couple of years. So from the fantastic chemists at Enamine, they've shipped us just under 2,500 compounds. And from this, we've collected nearly 10,000 data sets. From this, we've solved and publicly released nearly 600 liganded protein structures. And we actually have 300 structures that we're still in the process of refining. So if you compare these numbers to what's been deposited in the PDB regarding the main protease, which is slightly over 800 structures, we have nearly 600 structures ourselves, and there's an overlap of some structures which we've already deposited, with another 300 to come. One of the things that this project has really taught us, so the way our platform is set up, is we're really kind of looking at doing a single pass of each part of our experiment. We carry out the sample preparation, we collect the data, we do analysis, and then we publish. Whereas we're working a much more iterative cycle, and we're aiming for 1 weekly cycles to keep up with the assay teams. So just in this graph here, you can see in yellow, this is the weekly shipments of compounds. So you see they're very routine, roughly a similar number coming every week. And then in green, kind of teal, we have the data sets that we're actually collecting. And the nature of synchrotron is we don't run continuously throughout the year. We have dark periods where we have to go down for maintenance. These are just shown in these gray blocks. So you can see the data collection is a little bit more sporadic compared to the compounds arriving. But we have roughly kept in line and almost achieved that 10,000 data set number. Important thing we've learned from doing this project and trying to keep up with cycle time is really trying to cut down on some of our overheads and improve that cycle time, particularly when it comes to data dissemination. So we have this huge number of data sets of crystals, which we then have to align into a single reference frame so that people can have a look at the data and compare the ligand binding and the different conformations. But we really had to think about how do we curate this information? So how can we review the structures, flag any issues that we find with them? And then how do we annotate this with useful information for the design team so we can link them to different binding sites or different chemical series and include ligand quality annotations or site series annotations, all before depositing in our open web platform for Galaxis, and then later in other repositories such as the PDB. And I think my colleague Lizbe is going to talk a little bit about the data dissemination process.
Sorry, I'm struggling to share. Oh, there it goes. So I've also been in various moonshot trenches over the years, over 2.5 years. So I thought the easiest way is just to quickly show you what it actually looks like if we did a data analysis. And now my screen doesn't want to. Sorry.
I think I'm seeing Lizbe froze down. I don't know if worse comes to worse, whether she'd like to send you the slides, you present them, and she talks through them. If the share screen's not working on your side, Lizbe.
Or we just jump to the next next person and then feed them in if required.
Yep, Annette, Ben, I suggest you guys should start now, and then let's begin. Circle back at the end.
Sure. Okay. Let's see if I can get my screen to share. How's that? Okay, is it working from my side?
Perfect.
Okay, cool. So Annette and I are going to talk a little bit about where next. So everyone's given a great overview of what's been happening. We're going to talk a little bit about what we hope will happen and how we're going to take this forward. So this is the pathway to global equitable access. So Ed's given a great overview of the Medchem, and hopefully the plan is that we'll have a couple of molecules available from that work to nominate as preclinical candidates, probably around about February. So what you can see here is a timeline for the next year, next 2 years or so. We're about here. Here's Christmas 2021. And this is the plan that we're leading at DNDi using the Wellcome Trust funding that we've received for the Moonshot. So this is to take things through from the end of lead optimization all the way through to phase 1 readiness, which is all the way over here. So we kind of have about a 16, 17 month plan for the preclinical and the supporting work to go alongside that preclinic. And the way we're going to do this, hopefully, is we're going to pick around about 3 compounds coming out of Ed's selection, ideally quite different. So maybe a spirocycle, maybe a non-spirocycle, try to vary it up. So we've got a couple of different types of molecules going into the preclinic. And then we're going to start working on those. Going to do some early preclinical profiling, take these down to maybe 2 of the best compounds for some early exploratory talks, and then pick a final clinical candidate for the GLP work. And I'm aware that in a lot of the open science projects, particularly open science drug discovery projects that are going on, there's been some great examples in the past 5 or 10 years. It's quite rare for one of them to get as far as this. So this is a really great opportunity to think about, what is this? What is preclinical profiling? Here it's a bunch of lines on a Gantt chart, but what actually has to happen? So I'm going to take you through a little bit of this one by one. So first of all, when we have these 3 compounds, what are we going to do with those 3 compounds? So first thing, we've got to take these into early preclinical profiling. Very first thing we have to do is make 25, 30 grams of that, which is not that trivial. The Medchem team will have only been making 100 megs, 200 megs at a time. We have to quickly crank out about 25, 30 grams. And with that, we will use this to complete some in vitro toxicology. So start looking at hERG and CYP inhibition and really in some depth, secondary pharmacology panels, what else are the molecules hitting, kinome panels, these sorts of things. We also have to start really probing into the PK and understanding, well, we know the PK is really good when you dose it once. What happens when we dose it at higher doses? If we double the dose, do we double the exposure? If we increase the dose by 10-fold, do we get the exposure up by 10-fold? Where do we really sort of top out in the exposures? What happens if we do it on multiple days? Also, what about this? What about the PK in higher species? Ed was showing you some mouse and rat PK, but we also have to think about the other species that we might be using for the toxicology. So as Ed mentioned, some of the non-rodent species, and also understand how those compounds are then metabolized in each of those species, because we really need to make sure that the metabolites we're generating in our tox species are similar to the metabolites we're going to generate in humans. We can get some human MET-ID, we can get MET-ID in our different species and make sure that everything matches up, so we can understand that the toxicology packages we're going to create are going to be sufficiently robust to be able to indicate where we're going to be when we go to human. And also, we really have to make a robust human dose prediction. So Ed showed you the work earlier where the compound was predicted to be somewhere between 300 and 3,000 mg per dose, 3 times a day. I mean, that's quite varied. We really need to nail that down by doing a lot of detailed in vitro ADME to be able to get a much more robust PK prediction. And we'll also be doing in vivo efficacy at this point. So we do have plans to look at how these things work in mouse models. And we have collaborators at Mount Sinai who are going to be doing that work. It's not on the critical path. We're pretty confident in the mechanism of action here. So we know that if we can get compounds that show efficacy in vitro, in cells, via an MPRO inhibition, we're pretty confident we can kind of work out how those are going to work in human. But it's good to get that efficacy data in the mouse in parallel to help us drive this.
So that's this early preclinical profiling, which we're going to do on 3 compounds. But we also have to think, because of we want to do this as fast as possible, how are we going to make this on a much bigger scale? You know, making it on 100 mg is one thing, making it on 25 grams is another thing. Even more complicated, how do we make this on kilo scale? So we have to start thinking about scaling up and process research development. Is the medchem route, and Ed showed you a phenomenal medchem route earlier from the team at Enamine. Can we use that on scale? Is there something that we can do now that we're just making one specific molecule that will save us a little time? Are there experiments, are there reactions in that scheme that are not going to work on a kilo scale? If you're using certain pyrophoric reagents or certain exotherms, can you get around these? So we have to think about improving that route and scouting it so that we can get something that's suitable for really large scale, and thinking about optimizing each of the steps. And there's a heck of a lot of analytical chemistry that has to go alongside that, like an enormous amount of analytical work that needs to be developed to really understand impurity levels and the purity of the starting materials and the purity of the reagents and the purity of the output and everything. So a huge amount of analytical chemistry. And then we're going to make 500 grams of all 3 compounds ready to go into this exploratory tox. Also, in parallel to that, we have to think about the pharmaceutical development. So here, this is thinking about, well, can we make a salt? We've been using freebase for now, but if there is if there's a basic center in there that we can protonate sufficiently, could we make the HCl salt? Could we make the sulfonate salt? Can we make, what kind of salts could we use? Does that bias anything in terms of solubility? Does that improve our PK? Once we've got that nailed down, we have to start thinking about what the polymorph profile is. Does this compound exist as a single solid form, single polymorph? That would be fantastic, but unlikely. It's quite possible it'll exist in a number of different crystalline forms. How robust can we understand that, just so we can make sure that we don't have variability in our polymorphs? And then how do we formulate it to get it into the tox? How do we formulate it to get it into the higher species PK? So all of this work on those 3 compounds before we cut down to 2, so we'll eliminate the weakest and move 2 of them into the exploratory tox. Even though that 3rd compound is synthesized to the right scale, it's ready to go, we will let it drop at that point and we'll go into the exploratory toxicology phase. So this is when we look on 2 compounds. So hoping to do this the end of next year, looking at what kind of doses are tolerated in the 2 species we choose to go for, what the maximum tolerated dose is, running a number of days toxicology experiments in both of those. Huge amount of bioanalytical development for this. So we have to do all the bioanalysis in parallel to that. And while we're doing that, we're going to keep pushing on the process research development. So this is thinking more about the scale up. Can we improve the route further? How are we going to make this on kilo scale? Can we make 4 or 5 kilograms of this? Are there any genotoxic impurity profiles here? How do we make this into GMP standards, so good manufacturing process standard? And all of the analytical method development that's required to be able to get that GMP status. So all of this on 2 compounds, which is what's allowing us to go as fast as we can with these molecules in this plan. And then obviously thinking about pharmaceutical development for the tox studies. So what formulation are we going to use for the exploratory tox? What are we going to use for the GLP tox? And eventually we'll nominate, once we've got the exploratory tox for those 2 compounds, we'll look at them, holistic evaluation, which is the best looking molecule, and take this then into the final stages of the preclinic. So the GLP toxicology. So here we have to do everything to GLP standard, in vitro and in vivo genotox, in vitro and in vivo cardiovascular tox, respiratory tox, CNS tox, anything else that's been flagged up during the preclinic that we really need to nail down. Obviously the GLP 14-day tox studies, which we're going to plan on doing in 2 different species, and at the same time scaling this up for the phase 1. So making the active pharmaceutical ingredient, the API, 5 kilograms, GMP standard, ready to be able to go into the first in human studies in 2023. And all of the supporting pharmaceutical development that goes alongside that. How are we going to package it in? How are we going to formulate it for those studies? What the formulation is going to look like? So a lot of work that has to go in. And in parallel to all that, and Annette's going to talk about this in a little bit, but we're also trying to think about, well, who's going to take this further down the line? DNDi are helping coordinate this at the moment, everything up to phase 1. Who is going, where is this going to be developed, and who's going to be making this on huge scale if we want to get this to patient? And this is something that we're actively talking to people about. So we're coming up with a long-term strategy from a business development perspective. Who are those future development partners going to be? Who's going to help us make this on scale? where are we going to do our phase 1 studies? So we're thinking about this at the moment. Which countries are the best, are the most well suited to running a fast phase 1 study? We need to know that so that we can compile a regulatory dossier, whether it's an IND for the US or is it going to be a CTA for the UK, something like that. And so we can compile that regulatory dossier ready to submit as soon as we can. And all of this needs to be coordinated, and there are a heck of a lot of contracts and a lot of studies that have to be planned. So I don't know how many of you are used to seeing Microsoft projects, but this is a snapshot of about 50% of the Microsoft projects planned. So this is, this is the complicated Gantt chart that this is a much simplified version of. This is about half of what has to happen in the next year and a half. So a lot of work once we've got those molecules coming out from Ed. Annette.
Okay, thank you very much, Ben. I'm Annette. I work at the University of Oxford, and we here at Oxford and also at DNDi have been thinking about what will happen once we eventually hit phase 1 and then also phase 2 and 3. So we're kind of trying to come up with a clinical strategy at the moment, and we're actually incredibly grateful to Pfizer, because Pfizer has been amazing with publishing what they have been doing with their molecule. And we also obviously have the clinical data from Pfizer, and because we're targeting the same protein, we are getting an idea of what it may take to take that into clinic. We haven't got the same machinery as Pfizer, so we're looking at longer timelines, but there are some great learning points. So there are basically 2 options. By the time we kind of end phase 1 and are going to start our efficacy studies, Pfizer has skipped their phase 2A study, which is basically a proof of concept study, and they're based their 2-3 study, so their kind of big efficacy study, straight onto the data from PK/PD. So that is exactly there between where Ben's pointing at. So they kind of ran their phase 1, went straight into a large phase 2-3 study, which then was terminated very recently because of the kind of amazing efficacy that they have seen with Paxlovid. And that gives us confidence that we can model our approach onto that. So if there is still virus circulating in the population by end of 2023, which we are currently assuming, because there are still access problems, and we assume that there will still be access problems for drugs and vaccines at that time, there will be an opportunity to run our phase 2, phase 3 trials, and we will have to monitor the situation. And unfortunately, at the moment, it looks like our target area for the clinical phase may be in low and medium income countries, just because of where the kind of highest vaccine and drug access is going to likely be. The situation may change, but we think that this is the most likely scenario at the moment. We're considering an additional arm for virologic efficacy, and we will try to follow a regulatory strategy to get the drugs to the people that need it most as quickly as possible. So that is going to kind of drive the decision where we're going to run the trials, but also where we are going to register the drug in the end. There's the kind of ideal option that the pandemic has stopped by then. There's no endemic virus anywhere on the world. And in that case, we could just stop and stop after phase 1, or run a phase 2A on small patient numbers, just to demonstrate the impact on viral load. And that has been done for another antiviral drug that is currently in kind of approval or late stages, pre-approval, which is molnupiravir. And they used virologic efficacy as an endpoint. So it is possible to run that, and that is what we would choose to do. And probably in early 2023, we'll evaluate the situation and come up with a definite decision on which strategy we are going to take a year down the line. And Ben, do you want to move to the next slide? So you may ask, why are we still pursuing this approach? There's Paxlovid around, there's molnupiravir around, and there are other compounds, for example, the Shionogi compound coming out soon. We're expecting data on that in December. Why are we pursuing the moonshot? So we still think that there is a good argument to keep going. First of all, we haven't treated the disease, and we haven't controlled the pandemic until we have controlled it everywhere. That's kind of a very important access problem. But we also do know that we are going after a very clinically validated target, and that's thanks to Pfizer that we know that now. But what we are aiming for is an improved profile. So we would like to have something that can be given as a standalone therapy, so without any PK enhancers, with a safety profile that is suitable for women of childbearing age. For example, molnupiravir is very unlikely to ever be given to people of childbearing age. And also, if you have more drugs, more drug opportunities, you can kind of tailor your therapy and potentially limit the risk for drug-drug interactions. And that is particularly true for Paxlovid, for example, that has to be given with ritonavir, where you have a higher likelihood of drug-drug interaction simply because you also have to give ritonavir to get the exposure that's needed. The other thing is we are not going to have a good idea of what is happening in terms of escape variants, which is kind of further down the slide. We think that, or we know that our compound looks quite different to Paxlovid, and with that we are enhancing options for potential combination therapies down the line, but we may also be targeting different kinds of escape variants that will for sure turn up with time. We know that already with kind of the new variants that are currently occurring. We also do think that we are structurally different. So with non-covalent targeting, we may avoid risk of sensitization over multiple courses. This is something we do not know that yet, it's a suspicion. But what is a very important point to us is that we want to address global equitable access by introducing competition to the marketplace. And that takes us to the next slide. Which basically just summarizes our kind of main strategic ambitions for downstream development. What we really want is a new anti-COVID oral therapeutic that is available at affordable costs. We want to maintain the compound IP3. We also kind of see this as a bit of an experiment to kind of see whether we can get this drug to the market. We are prioritizing LMIC needs and access to treatment, and we are aiming to work very closely with LMICs, the WHO and ACT-A, who are also funding us through the Wellcome to get this compound to control this pandemic or potentially prevent future pandemic or global outbreaks. And then finally, this is just very quick thoughts on potential business strategies. And Ben has already mentioned that for the kind of big scale development, we all need a very, very capable partner, someone who can produce the drug at extremely large scale and distribute it. And ideally, we would also get someone that has regulatory experience with new molecular entities. But we can also envisage, so that would be potentially a large pharma partner, but we can also envisage working very closely with a generic manufacturer because they have very similar distribution and large production networks. So we are exploring these different options at the moment, talking to multiple partners, and there is actually a lot of interest, which is good, but we're very, very early in this kind of stage, in the kind of business development stage, and DNDi is leading us through that, and we are, as the Moonshot, incredibly thankful for their support in this particular stage. And this brings us to the end of my presentation. I just want to kind of mention, while we are kind of loading the next set of slides, I just wanted to mention to all the people that have been running antiviral assays for us, you haven't been in the trenches today for the presentation, but we couldn't have done any of this work without you. And we all know how incredibly work intensive these assays are. So a big shout, and if we are still here by the end of next year, you will be in our presentation. Alpha.
Yep, thanks, Annette. And on that note, hopefully by mid of next year, we will be able to launch a broader vision that takes Moonshot to the next milestone and carry the vision of Moonshot and expand our platform to actually going beyond reactive drug discovery to preemptive drug discovery against emergent pandemics. So in this presentation, I will talk about our vision for the future of Moonshot, launching the AI-driven structure-enabled antiviral platform, which we have submitted to the NIH for funding, and hopefully we'll be able to realize this vision and launch a evergreen platform for antiviral drug discovery. So first, perhaps taking a step back and reflecting what we have learned from Moonshot that inspired this vision. I think one thing that we've learned is that open science has a very powerful networked effect. Over 100 articles or preprints or projects have cited Moonshot's data that's straight from Google Scholar, the source of all knowledge. And openness actually facilitates collaboration of industry by not having MTAs, by publishing everything online, and our compounds are actually orderable from Enamine, means that we can collaborate with industry, collaborate with academia in a way that is very, very facile and easy. So there's a huge network effect, and the map that Aaron showed right at the beginning shows how open science allows to create a community in pretty rapid speed. The second learning is that most funding course, especially towards the beginning of the pandemic, or even to some extent now, underestimated the duration of COVID. You can clearly remember UKRI back in the UK saying that, you know, pandemic research should have an impact horizon, I believe 3 to 6 months. We are now, 2 years into it and still not over yet. And overestimated the duration and cost of a tech-enabled modern antiviral drug discovery campaign. And I think the Paxlovid breakthrough by Pfizer is absolutely phenomenal. I think that showcases how rapid drug discovery can be done. I think that's a true breakthrough in medicinal chemistry, the team at Pfizer. And it is actually possible with modern technology to discover and complete a clinical trial in time to impact a pandemic. I think that story tells us how, we shouldn't think of small molecule drug discovery as necessarily this endeavor which we should not start during a pandemic, but it is actually worthwhile to take a shot at it rather than waiting and waiting and waiting. And in fact, Moonshot, we reached the preclinical stage with $1 million and in 14 months from a cold start. And we are very grateful for follow-on funding, which allows us to now go into late lead optimization IND enabling studies. So these two learnings are nucleated in a vision that we can actually achieve a pipeline of antivirals against future pandemics if we start thinking about antiviral drug discovery as a public good. I mean, obviously there are commercial potential antiviral drug discovery, and we do agree that there needs to be a commercial efforts against that as well. But antivirals and pandemics is a global humanitarian crisis and concern that should be augmented in addition to commercial effort, state effort, to rethink antiviral drug discovery as not only a commercial product, which it totally is, but also a public good. And in fact, if we look back, this figure really somewhat saddens me, that if we test our compounds against SARS-CoV-1 back in 2003, the epidemic in Asia, we find that actually the potency against SARS-CoV-2 main protease of our compounds are equivalent to potency against SARS-CoV-1 main protease, which means that there is actually no reason why we couldn't have done Moonshot in 2004 with the last version of the main protease right after the 2003 SARS pandemic. And you may argue drug discovery is slow, but with 15, 16, 17 years, we would have crawled towards a main protease inhibitor, even if we were to bootstrap right after the 2003 SARS pandemic. So it is a pandemic of neglect rather than a pandemic because we don't have this science. And we wrote this op-ed to argue the case that it would be great if we can stop future pandemics by developing a deep pipeline of antivirals. And that really nucleated the vision of thinking about this not only as a campaign against the main protease, but also as a broader vision of how can we create a platform that can develop a pipeline of antivirals. And we think about this scientific vision in 3 different parts. First is aiming for the right targets. I mean, the viral genome is there are quite a few proteins in the viral genome. We all go after the main protease because it is a validated target. But there are so many more proteins out there, and as Annette mentioned, resistance is always an issue. So how do we aim for the right targets? And then the second piece of the jigsaw is discovering the right molecules, and that pertains to using technology-enabled medicinal chemistry, machine learning, computational chemistry in conjunction with high-throughput structural biology to find the right molecules. And finally, really engineering access and engaging the right community right at the beginning of the program. So these three pillars forms our thinking when we are nucleating this new idea of ASAP, which is our proposal to the NIH. So first, we focus on viruses with significant pandemic potential and disease burden. So this program, this pitch that we have submitted to NIH will focus on the coronavirus for obvious reasons. Flavivirus, which is the causal agent of dengue, actually infects 400 million people a year. And even in the US, southeastern US will become at risk in 2050 due to climate change. Mosquitoes will start coming to the US. And antibody-mediated disease enhancement hinders vaccine development. And then we're also going to look at piconavirus, because even the current piconavirus, AViDD68, causes polio-like permanent paralysis, like polio. And there are actually recent outbreaks in the US. And the diversity of piconaviruses renders vaccination coverage impractical. So these are the three viruses that we as a consortium plan to focus on if we were to be funded. And as I alluded to, we have three pillars: the right target, the right molecules, and the right patients. And so for target identification, we really want to take resistance into account. And we do this in several ways. The first is robust phylogenetic analysis to identify conserved binding site across viral strains so that we understand which targets are broadly conserved across different viruses. And then we put in deep mutational scanning to explore sequence space more extensively than natural evolution to identify binding site envelopes that we should target. And finally, novel next generation so-called dominant target that when complexed with compounds that inhibits it will create basically toxic byproducts for the virus. So it's a self-destruct mechanism. If we inhibit this target, the protein substrate complex would itself stop further viral replication and therefore suppress the emergence of mutational variants. In terms of the right molecules, the Diamond Chem team, as Daren alluded to, has done actually phenomenal work over the past year, executing fragment screen over and over again against a large plate of a coronavirus target, showing that this fragment-enabled high-throughput structural biology technology is not a single one-trick pony against main protease, but it's a platform technology that can be scaled across the coronavirus genome, but also, we believe, across the viral genome of piconaviruses and flaviviruses. And finally, engaging the right community right from the get-go. We are very delighted to have the DNDi on board, which has a proven track record in therapeutics for neglected disease and neglected patient groups. We want to ensure equitable global access right from day zero and have a pro-public health stance towards intellectual property. And finally, keeping open science to ensure that all data is immediately available to the research community, therefore creating not only a pipeline of antivirals, but also an open knowledge base for antiviral R&D. A rising tide lifts all boats. And this is a schematic of what we have put together as a consortium, combining 5 scientific pillars, going all the way from target identification, led by Carla Kierkegaard from Stanford, to target enablement, thinking about how do we create robust assays and create robust fragment screens, led by Frank, and then fragment to lead and target validation and covalent target strategies, led by myself and Nia, lead optimization, led by Ed, and preclinical development, led by Ben and Annette, and several scientific cores, data infrastructure, structural biology, biochemical assay, and antiviral assays. And we are very delighted that Dr. Fauci launched a program inviting applications for so-called AViDD centers, US Centers for Antiviral Drug Discovery, and we have submitted a proposal as Moonshot to this program, and we hope to be able to be offered the opportunity to extend and augment the vision of Moonshot from a reactive program against the coronavirus to a proactive program that safeguards our future against emerging pandemics. And with that, thank you very much.
Great. I think before we launch into the Q&A, we'll bring Lizbe back in because I think she's managed to rejoin us all. And Lizbe, you can take it away with the slides you've obviously put a lot of time to put together.
So I'm hoping it's not going to freak out. So I had one job to do, and that was to explain or just give you a practical point of what Daren was saying, how it looks in the lab. And I think I've already done a good job. You now know how it looks when graphics stall, when your bespoke software freezes on you and you have to restart your computer and delay everything because things are just not working as it's supposed to do. So that's the bad side. So on the other side, Daren basically, when he ended off, gave this overview slide of how we do the data and go and curate it. And I thought I'll just actually show you what this looks like if I walk in and log into my computer. So all those nice crystal structures and compound IDs for us we pull up an interface that looks like this, which called Extreme Explorer. And from this, we basically run most of our processes. This nice colorful blocks in the bottom is the process, and Daren actually takes care of everything up until after hit identification and placing the ligands for us. And then where I come in with the data processing is the structure building and refinement. And this part which is the time sink in our process, and this is also why I put the clock here. So in a very, very simplified version of what we're doing is we look at all the structures. So for those who don't know, all these blue and green webbing is basically the electron densities, and this orange thing striped through is the enzyme structure, or in this case where my mouse is would be the ligand. And what we need to go through is we need to go through all of this, look at things that doesn't make sense. So as you can see, this red dot doesn't actually fill this space. But also if it's green, then it normally means something's missing, so something needs to be placed there. And if there's red density, you can see in the back, it means something's there that shouldn't be there. And what we now go through is a lot of iterative cycles of trying to fit things in here, fix this, so we get to the best possible model that the med chemists can trust. Once that is done, we push it out to a program called XChem Review. So if I've refined the structures, which would be the case, then somebody else independent from me will look at this and say, yes, what you've done here makes sense, or it doesn't make sense. And they basically have this, you can release the structure to Fragalysis, Ed and the Medchem team go look at it, or they can send it back and say, no, you need to do more refinements or more experiments, or they can reject it. Reject doesn't normally happen at this stage because we have a good process in the front. And while I would like all of this data transfer to be one way in this direction, there's actually probably an equally big arrow going back sometimes, just saying, just tweak this, just change this a little bit. But why I'm showing you this is just to say that by the time it gets to Ed and the Medchem team, there has been a lot of process in place to check to the quality and to make sure that what we put out there is actually reliable. And then, of course, as soon as it's been approved in XChem Review, it gets pushed out into Fragalysis. And Fragalysis is our public viewer site that you all would know and love. If you just actually Google Fragalysis Impro, you will be able to launch into this page. And from here, you can see all the compounds, the hits, how it fits into this. And if you play a bit more, you can actually also see how the binding interactions work. And then, yeah, that is all I had to say. So I'll stop there.
I think Frank has kindly offered to jump in on the Q&A, given we lost John. So Frank, over to you.
Yeah, thank you very much. For those that remain, there was a few questions in the chat. I'll just go through them one by one. So I don't know which direction they're going in. Peter Kenny asked, are there specific plans to exploit catalytic cysteine as a molecular recognition element? So potential benefits if we need to deal with mutants and SARS-CoV-2. I suspect for this current program, Ed can jump in if he says, but I think the general view is that we've gone so far down the lead optimization that that's not something we can currently do. with the current amount of funding. But if we get the AViDD grant, the ASAP that Alpha discussed, this will be completely in scope. These are exactly the considerations, and it's obvious, looking at what for instance, Paxlovid are doing, that these are things to pursue. So it's been interesting for me to watch how one has to discard ideas as you run down the track, and of course we've discarded many other ones, and how important early decisions are about whether or not one keeps going with covalentizing it. So there's a brief answer. Will Ward had asked, have you looked at human plasma protein binding? Ed, I don't know. I know you had some slides up. Do you want us to add anything to that question?
We have done quite extensive profiling in both human and rat, and to date they mirror each other pretty well. But we continue to monitor that, obviously, because the point that Wal brings up is exactly right. We don't want to end up producing a rat-only drug, or even something where the in vivo experiments we do in rodents are not reflective of human. So we have it in progress.
By the way, if your question is answered and you want to add to it, just chip in. Alpha, maybe you take this one. John Spencer asked, there was a preprint with about 150 plus authors. Did it ever get submitted? And if so, is there any progress?
Thanks, John, for the question. We have submitted this to bioRxiv. We are still compiling the final pieces of data to prepare for a journal submission. We hope to do this in the next month or two. So early next year, it will be submitted to a journal.
I should say that there's been quite a lot of iterations on what exactly is the scope of what should be published, because as you of course, to take a snapshot and then generate the paper out of it, that's not been a simplest of decisions. One for Ed. Vera Omics asked a few questions, but basically, what do we think the impact of the Omicron M-Pro mutation will be? It's P132. It's a proline that 132 that's mutated to histidine.
We have looked at this. It's quite remote to the active site that we are currently binding in, the P1, P2 sites. Obviously, that could induce conformational change, so it is a risk. So the current proposal is to go ahead and actually produce the clone where that mutation has occurred, just the enzyme, not obviously the virus, and then actually measure that. So at the moment, we don't consider it a severe risk, but we're going ahead and we'll do the experimental work as we go forward. So within the current understanding, we should be okay.
Okay.
We will also test our compounds against Omicron, so in cells. That's what's currently happening as well, in addition.
And Chris has just added that the Pfizer impregnated maintains its efficacy against the Omicron in protease assays, which not that surprising, but also reassuring. Ernest Williams had asked, I'm curious about the accuracy of the absolute binding free energy simulations and how they informed compound design. So John was going to answer this, but he just left the answer to this question. He says, Vincent Wells is working on a manuscript about the absolute free energy calculations, and the Chodera lab is diving more deeply into the quantitative comparisons of accuracy for relative free energy calculations. So it's one of those resources that we're very happy to have come out of the Moonshot as a side product, and so it's active work, if that helps. I suppose just contact John directly if you want more information on that. One for Ben, maybe. Peter, can you ask, what are the plans for backups and follow-ups for the clinical candidate? The things we have to leave by the wayside. Is Ben still here?
Yeah, I'm still here. I think I mean there's a wealth of science here available in the series, and I think that the opportunities for backups or second generation, third generation is still there. It's not covered by the welcome grant. The welcome grant is first in class, quickest. What's the best molecule we can get? Alpha talked about AViDD. It's quite possible that we'll have the opportunity to look at second or next generation compounds within that scope should we get the funding. I'm not sure if anyone else wants to add anything to that. Ed, perhaps.
Well, we have live discussions with NCATs in terms of where they can put chemistry effort. Clearly, it will be good to have backups. It just depends when they're able to engage with that.
I think it's ultimately a funding question. If we can convince someone to invest in these questions, and yeah, it's not the not the new problem. So we had a question from John Spencer, probably for Ed. Is your quinoline 2 position a metabolism problem? Can fluorination or CHF2 be of use?
We don't believe so at this point. However, I will be in touch with you, John, because we're having discussions about introducing CHF2s in other places. So I think I need to give you a call and discuss the synthetic chemistry options there. So I'll be in touch.
Another one for Ed, although I guess others could take it as well. Garrett Morris asked, how can knowledge of how the 11 substrates bind to Mpro help in developing the latest compounds? For instance, the cyclopropanolnitrile group is sitting in the P3 site, so maybe amino acids that sit out there might point to new derivatives. Maybe I'll start on this one, Garrett, because we've been having quite an active or trying to figure out in the first instance if these interactions in the P3 site are productive for the potency, or if the P3 site is a little bit incidental. It's got more to do with conformations of the molecule itself. They don't seem to be crucial interactions for binding, but the preservation or the conservation in that site isn't that high. Of course, it's the P1 and P2 sites that have the highly conserved residues, and we think that the compounds are really mainly exploiting the substrate envelope, so the bits that can't mutate away, which is why we are feeling quietly confident that that the virus will struggle to mutate away, or at least that's the way people think about certainly protease inhibition, or how to preserve or make things mutation robust. So you stay within the envelope where the virus cannot mutate away. So those substrates and comparing them has been quite important to at least give us a sense of where we should stay. Hey, do you want to add anything?
No, the only thing I'd add was, unfortunately, Near London's had to sign off, but in his group, they're doing some really interesting screening work about screening a large number of potential substituents in that site, basically derived from glycinamides. So we'll have much more data about how the actual potency varies. But I think it's a good prompt to us to go back and look at that knowledge. So I appreciate the prompt.
And Garrett, I see you just added that Celia Schiffer works on it. In fact, she's the one that has been really putting that term substrate envelope to prominence, and it was important way for us to think about this as well. Are there any other questions? I see nothing else in the chat, but please put up your hands or pipe up for anybody. No, going a little bit over time, so I assume it's been a long session. I'll just hand back to Aaron then.
I think that's good. Thanks so much, everyone. Again, if you do have questions that come to mind, please just email at postera.ai. Again, we try and keep everyone updated on Twitter, but if you have separate questions, send them to that email. Otherwise, apologies for running over. Fantastic to see everyone. The recording will get posted online somewhere at some point. And with that, just wishing everyone a fantastic Christmas, New Year, and see everyone in 2022. Thank you again.